Good morning, everybody. We're going to get started today. Final lecture of a series of 4 on drug discovery and development. Yesterday I talked about monoclonal antibodies as a new type of medicine available for development, a little bit about some of the technologies and the pharmacology. Today I'm going to switch it up a little bit and talk more about how we merge strategy, understanding unmet need, and use of technologies and the timelines for development. I'm going to do this as a case study because I thought it would be more interesting and I think easier to follow. Just as reminders again, we have questions. There are participants online that are watching in today, and then of course we have our students here in the class. We'd like you to also fill out the survey questions to see how we've done in actually delivering messages to you. And of course, there's always the course evaluation. So again, the high-level objectives for today. I'm going to present a case study, an actual drug development program going on to try to illustrate some of the key things that one has to consider while developing a biological drug. It begins with actually the drug concept, understanding the competitive landscape and the strategy for success. How do we use technologies in an optimal way, not just to get at a better solution in terms of an actual drug-like molecule, but the ability to use freedom to operate and get new patent position to support this drug as a new thing. There's the preclinical testing. What kind of tests can you do to actually demonstrate efficacy? And then you go on to actually generating new intellectual property, manufacturing, IND-enabling studies. And then depending on where you're at as a company, or if you're going from the academics into merging with a biopharma, what is the business development and actually the partnering considerations to actually move this forward? Because clinical development is very, very expensive, and you need to either raise cash, partner, or license at a particular time. So for today, I'm going to talk about a case study. This is really the best-in-class anti-CD22 drug conjugate for the treatment of B-cell cancers. A little bit about the CD22 superfamily. It's a member of the Siglec family of proteins. CD22 is a transmembrane protein. It's actually involved in signaling and regulating B-cell function during development of the immune response. And it's closely related to a number of molecules, and they have very similar structure where they actually have a binding head that binds to sialic acid containing ligands, and also several immunoglobulin motifs. And you can see this type of more or less motif that's covered across this family. There's another molecule, CD33, which has actually been the target of another antibody-based drug called Mylotarg. And actually there's another protein, Siglec-15, that is currently being developed as an antibody medicine for treatment of bone loss. And it's not clear exactly what the function of these proteins are, but they appear to work by regulating key receptors involved in differentiation of a number of metabolic lineages. But they bind something that's in the microenvironment that these should actually develop in. And I think it's a way of controlling to make sure that development of these very important cell types occurs in the right microenvironment to provide function across the board for the variety of cell types that it's expressed in. So just a little bit about CD22 biology. Again, it's a transmembrane protein. It appears to affect the regulation of the antigen receptor during B-cell development in context with antigen. And it's doing so by actually regulating binding to some motif, and upon happening to that, it can either interact with the B-cell receptor or not. So it can have actually positive regulation and negative regulation properties to it. It becomes phosphorylated on tyrosine residues, and then different binding proteins can interact with that intercellular domain and cross back and actually go work on the downstream signal transduction. One of the most important things I think I really want to tell you about that there was a very important antibody, a mouse murine monoclonal antibody called LL2 that was developed really during the late 1980s, early 1991. And it was found that when that antibody binds to CD22, it induces phosphorylation and actually can negatively regulate B cell differentiation, growth, downstream of the signaling transduction apparatus from the B cell receptor. And this had an effect to actually decrease B cell growth and development in vivo, and actually could block the growth of certain lymphomas and leukemias that highly expressed the CD22 protein. So CD22 is expressed really on a very discrete subset of the B cell lineage during differentiation, the mature B cells and the developing pre-B cells. And that's in sharp contrast to other B cell specific surface antigens like CD19 and CD20, both of which have been targets for therapeutic antibodies. CD20, of course, is a target for a very well-known drug called rituximab, which is one of the frontline treatments for treating non-Hodgkin lymphoma. And it works very well, but what you can see, it actually depletes a number of cell types within the B cell lineages. And CD19 is just a target for a recently approved drug just last week from Amgen, which is a bispecific that binds to CD19 and also brings in CD3-containing T cells that can have cytotoxic effect in lymphomas. So these are very important. So an important concept is that there are other drugs out there. There's standard care. There's also an anti-CD20 and anti-CD19 drug, which are all marketed. But there's really problems and unmet need in that they don't eradicate all the lymphomas together. CD20 targeted by rituximab works extremely well, but only in about 33% of the patients. So the rest of the patients, there really are no effective cures. CD19 similarly works on a very select subset of types of leukemias and lymphomas, and it doesn't. So there's still unmet need for treating very important lymphomas and leukemias by targeting surface antigens and using this in combination with standard care, including chemotherapy drugs. So CD22, as it turns out, is almost universally expressed in most leukemias and B-cell lymphomas, including the most common ones, for example, like follicular and mantle cell and DLBC. And it's very clear that in tumors that are targeted by CD20, rituximab, that it's not just the loss of CD20 that causes the failure of a response in those tumors, and it's not really clear why. There must be some downstream mutations. So by targeting another antigen, you can actually think about using the combinations of different drugs and looking at patients that have relapsed from the rituximab treatment and coming in with other drugs to bring in more effective cures. This is a little data here. I'm not going to go into it, but for those people who like to read up and understand more of it, there's a whole list of literature about the expression of CD22 across the board in B-cell leukemias. And the other thing that's interesting about it, it's really not expressed in any other normal tissue, so it's almost a very specific and selective target. One can think about actually going in with a very potent anti-CD22 that's armed with a warhead and getting a very good effect, therapeutic effect on lymphomas and leukemias, and not really harm other body tissues, although there will be some other depletion of normal B cells, but that will be minimized because of the very select expression pattern during B cell development. So this is the kind of the drug concept, actually a new CD22, an improved CD22 antibody that's armed with a drug conjugate. This is just a little bit to tell you about what are some of the CD22 molecules that are being developed in the clinic. Starting from the right-hand side, there's a drug called epreptuzumab that was actually derived from the mouse monoclonal LL2 that was humanized and optimized, and it's actually being used to treat lupus patients in the clinic. I'll talk a little bit more about that in a second. It's in the latter stages of phase 3 development. The results should be posted in the first quarter of next year. It is a naked antibody, unconjugated. And then there's an antibody in phase 3 called inotuzumab. This is an antibody drug conjugate by Pfizer, which contains an anti-CD22 antibody that is conjugated to calicomycin, a very potent cytotoxic drug that targets nucleic acids. As I'll show in a moment, actually there's some problems with that. Because of the instability of the conjugate, there's actually a lot of free drug that's leaking off, and you're actually limited by how much drug you can give, even though it's having some potent effects, it's actually being de-emphasized in phase 3 and might not continue. Genentech has an anti-CD22 antibody that's armed with basically a auristatin E conjugate. This is a very potent microtubulin inhibitor, and it's now in phase 1, phase 2 studies, and it's having very interesting effects. They've showed proof of concept actually in phase 1 studies, but it appears potency of this antibody may limit its actual utility. So what we did is actually came up with a concept in saying we're going to take an improved CD22 antibody, make it very potent in terms of affinity, look for very high surface binding and internalization. And as an antibody, we have two choices. We can use the naked antibody for development in autoimmunity, and we can make an antibody drug conjugate for use in oncology. So we took these two paths with this, and I'm going to talk mostly about the antibody drug conjugate side of this and how we kind of more or less use technologies to develop a more potent antibody. So this is the overall kind of drug concept, and I'll talk a little bit more about the strategy. First, let me just tell you a little update on eprabtuzumab. It's going on in development. It's having some very interesting effects in lupus patients. If you look at a variety of indices that measure a whole score or assessment of how disease is progressing in these people. In phase 2, they showed efficacy, and currently there are 2 phase 3 studies ongoing, the emblem studies and embody studies. And these results should be out in the first quarter of 2015. So as a naked antibody, a very important need or unmet need in autoimmunity. There are other potential uses of this in other B-cell autoimmune diseases and potentially also rheumatoid arthritis. Inotuzumab, this is the Pfizer anti-CD22 clindamycin conjugate. The upshot here is actually it looks very good in terms of targeting cells, particularly in Rituxan-resistant lymphomas. The issue has been because of the leakiness and the breakdown, they've been having some serious adverse side effects that actually limit the dose that you can deliver that. And these have to do with effects with thrombocytopenia, anemia, obviously in the bone marrow, and there's other side effects which have actually hampered this development to the point where this particular drug is not being used. And I should note that the conjugation technology is kind of what I view as the first generation of antibody drug conjugate technologies. Clindamycin is a very complicated molecule. As you can see it up there on the very top left, it's actually made as a natural product. There's no synthetic solution for making this in the laboratory. And the actual drug linker that used is not very stable and falls apart. And even the vial as it's delivered, it's felt like half of the drug is actually free drug that's not within that. So as you dose it, you're not getting targeting specifically to CD22. You're getting this more or less systemic side effect, but still very interesting and promising results. CD22 MMAE, there's not a lot of results published on that. This is Genentech has developed an IgG to CD22 that's conjugated to the auristatin E. This is from Seattle Genetics. This is a toxin which actually causes the breakdown of the microtubule apparatus, stops cell division, and leads to the cell death. It's had very good effect in patients, but they may have potency issues. And of course, there will always be relapse and resistance developed to microtubule inhibitors. So we felt that there's clearly going to be an angle to bring in a new drug, particularly if you use a new type of mechanism on the payload side. So this was our kind of basically drug concept, new best-in-class anti-CD22 antibody that if you combine with a more recent, more potent linker toxin combination, you can actually develop a superior drug for the oncology market that could be merged into the clinic by looking at resistance or relapsing patients, and depending on the efficacy, it could slide up potentially to be first in line for treatment. So this is how we kind of viewed the world as we were going forward in this project. And we really looked at what was out there, and there was EMAB out there, which is being developed by UCB as a naked antibody. There's inotuzumab that's out there that had very good effects, but being de-emphasized, probably they're not going to pursue this anymore in phase 3 studies because of the side effect profile and the inability to actually dose it high enough to get really complete cures. And then there's the CD22 MMAE. They used the actual conjugate, again, that is targeting the microtubule apparatus. And so we said we're going to build an antibody where we have more potent binding and internalization activity based on engineering of the variable domain of the protein. And we're going to bring in a new type of payload. We're not going to go for something that targets the cytoplasm and the microtubule. We're going to go target things that are in the DNA. And there were some nuances to why we did that based on this antibody and how it went into the cell and how it localized to a very close perinuclear localization. I'll talk about that. But the idea really about here, we said, look at EMAB, that came from a mouse monoclonal a long time ago that's still out there, and it's kind of off patent. We decided we're going to start with that mouse monoclonal antibody, engineer it in a way so it's more potent than any of the other molecules out there, but the sequence is different, and we've used technologies to give us complete freedom to operate and can patent an actual new drug. So instead of making new monoclonals, we started from existing one and took this, what I call this kind of dry approach to getting a new molecule that has complete patentability, freedom to operate, and a different, basically, product profile associated with that. So this is how we look at things. We said, okay, let's line everything up and say, how are we going to be differentiated from the molecules that are out there? And we see the different properties that we want to look at and how we compared them across the board. We looked at affinity, and we saw the EMAB as a lower affinity antibody. It certainly can improve that. We talked about yesterday how by improving affinity, you can improve the pharmacodynamics of the antibody, and that's a very important aspect of getting superior effects in the clinic. There's inotuzumab, which had pretty good affinity, but we thought we can do better, and there's the Genentech antibody, which had about the same affinity as EMAB. So we actually derived an antibody that had much higher affinity, and I'll show you that affinity translated into increased surface binding and actually internalization. There's the mechanism of action, and we'll talk a little bit about the internalization in a minute and why that's important. In terms of the mechanism of action, we saw EMAB as a negative growth regulator for B cells. That was going to be very important for B-cell dependent autoimmune diseases like lupus, rheumatoid arthritis, potentially MS, and other B-cell dependent autoimmune diseases. Inotuzumab delivers cholinomycin. This is a DNA binder chelator. It actually blocks DNA replication. CD22 delivers auristatin E, a microtubular inhibitor, in a different part of the cell. And then VM101 was going to develop something that was distinct from the Genentech molecule. And we wanted to go after a different mechanism, and I'll tell you a little bit about the payload, but it targets DNA. Isotypes across the board, IgG1 for EMAB, very long half-life, very good manufacturing properties. Inotuzumab was an IgG4, shorter half-lives in the body, more difficulty in engineering. There's some nuances to IgG4s and why you want to use them. For example, the light chains can actually exchange with other light chains from other antibodies in the circulation, so there's some potential issues with that. That has subsequently been solved by genetic engineering, but when this molecule was made, that was an isotype that would probably not represent the best choice for going forward. Genentech, also an IgG1. We wanted to go with an IgG1. We wanted those long half-life properties, manufacturability, stability, et cetera, that are associated with this particular subclass of immunoglobulins. Linker stability, obviously we wanted to go for things that are very stable in the circulation so we wouldn't get breakdown and release of the toxin. We're not differentiated from the Genentech molecule in this case, but we are from the other antibodies. And the stage of development, clearly VM101 was behind the scenes, so it really had to be differentiated up front to come up with something that would potentially be used in the clinic. Target product profile, I won't go off in this in distance, but you know, when you really start to think about things, before you get going, you think about the clinical need, the technologies, and what are your design goals? What are you going to make something like? And we can come up with an actual profile so that when we're actually developing something, we can look at it and say, you know what, we've gotten to where we want to go. We're not going to keep working on it. We've hit these product profile design goals, and we're going to move on for those. And those, of course, are affinity. We have some affinity targets based on our pharmacology assessment, isotype, what our projected half-life would be, so this would control dosing and not only frequency, but in terms of the volume. Route of delivery, initially this would probably be an IV delivered drug. Hopefully we can get it to a subcutaneous injection format, and we talked a little bit about that yesterday and why that would be important. Proof of mechanism, safety, we wanted this to be predictable and manageable in the clinic, whether it was a naked antibody or an antibody drug conjugate. And then proof of concept, depending on the pathway, whether we were going for autoimmune indication or oncology, proof of concept. There are a variety of situations. For the naked antibody, we can go into, for example, psoriasis, do a very small phase 1 clinical trial, show effect, and then move forward for them. We had timing to IND, how long it would take to actually get into patients. Biomarkers were considered, how we're going to look at this clinic to see, in the clinic to see that. And then proof of concept, what endpoints, what we're going to look for in the clinic to show this is actually working and we're on track before moving forward to more extensive studies. And then of course always about later on how do we expand indications in the clinic by looking at other patient populations, et cetera. And this is our overall kind of goal we had drawn up before we actually did any work in the laboratory, okay? So this is a very important thing strategically, is to think forward, okay? I want to make something for the clinic, let's define and make sure there's an unmet need, that there's going to be a sufficient patient population to actually test this and get it to market. And then work backwards for what is the design goals you want to do. And that tells you exactly how you can go back and use the technologies and then what you can source. Do I have to make a new antibody? Can I take an antibody that's already existing out there and start with it, develop intellectual property on that and freedom to operate, and then go forward and move forward. And once you have that picture in mind, working back to the starting point, you can start to navigate through the use of the technologies, understanding what you need to develop, what tests did me run in doing that. So this iterative look between looking out to the horizon of where you need to be, going back to the beginning, helps you really steer a course that is going to be very efficient in terms of the economics and also in terms of time. So let's go back now and tell you a little bit about the selection of this compound, how we thought about it, and the strategy around the intellectual property, the use of technology, et cetera, and how we could meet our design goals. So just before that, I'm going to go to summary. The idea was we're going to take the mouse LL2 and engineer it going forward. And we did this in a way, and I'll summarize it in a few minutes in a diagram, but the idea was we're going to take this mouse monoclonal and actually determine how we could use it to get freedom to operate. And it turns out if we were to take the mouse monoclonal and first affinity optimize it and change the CDR sequences so they were far enough away from the original patented CDR sequences, we could generate new IP around that. So we figured out a way to do that, and actually we used technologies that were based in China where there were no patents on the CD22 or LL22 antibody, made some iterative changes, improved the affinity, and then we went on to humanize it. We used a humanization technology which was novel and did not interfere with other technologies that were out there, and we came up with a brand new molecule, and that molecule is actually patented. The composition, I'll talk about it in a minute, and it's actually being partnered and going on to development. So by knowing what was out there and then understanding the strategy around the IP and then getting to specific design goals, we could actually save a lot of time. It takes a lot of time to make a new monoclonal, screen them, test for biological activity. One was already out there existing, the sequence was available, and we moved on from that. So by doing that, we really came up with 20 different candidates that we obtained from the murine LL2 monoclonal antibody. We selected the sequences based on the fact that we knew these were divergent enough from the existing IP that was out there. We looked at the affinity, we looked at surface binding and internalization by a number of assays, and I'll show you one using confocal microscopy. 10 of those went on to some very sophisticated internalization studies to make sure we're getting surface binding. And the reason why that was really important is, as an antibody drug conjugate, you want it to bind, internalize, get into an endocytic pathway, have the linker be cleaved within the late endosomes, which are acidified, and then released into the cell interior. In this case, this happened in an area that is very close proximity to the nucleus, so we wanted a very potent nucleic acid inhibitor attached to that antibody. We selected 3 clear leads based on this activity. And the leads all had very potent surface binding and internalization. And more important, we started to look at the actual sequences and say, aside from just being novel intellectual property based on the composition of the sequences, we started to look for particular residues which tend to give you problems in manufacturing. These are oxidizable residues, for example, asparagines, cysteine, methionines, where if you have a number of these things can get oxidized and change activity, give you a variety of different species during the characterization, which tend to be problematic. Certainly free cysteines, you can get aggregation and cross-reactivity. So we selected all of those out and went for things that we had best sequence composition just for manufacturability, not for bioactivity alone. We tested, can they be expressed at high levels? We actually put these clones into CHO cells and compared them to the expression of a marketed drug, for example, Herceptin or Humira, and saw that they all could be expressed at very high levels. They were well behaved. They didn't aggregate. You could be concentrated. So all these kind of physical properties of the antibody became very important in the downstream selection. And then we came up with a clear lead and 2 backups to take forward. So in case there were any problems with one, we could go forward with other ones. And that's kind of the strategy for picking out a clinical candidate through all those different processes. So it's filtering. It's not just activity. It's about a number of other things that have to be taken into consideration. Because up to this point, it's relatively cheap to develop an antibody. I say relatively cheap. We're going to go over the cost in a minute. But it's basically relatively cheap. The next steps are very expensive. Manufacturing, clinical development. You want to make sure you get it right at this point, because if you don't find that out until later on, you can spend a lot of money and have to start over. You've not only lost money, the economics of that, but you've lost an incredible amount of time, and this is a competitive area where someone else is going to be fast following if you don't. Again, here is the approach to humanization. We talked about this a little bit yesterday. We used the dry approach. We took a sequence, we basically altered it, and then eventually went into the humanization process. And the humanization process that we used was different than the standard CDR grafting that were covered by various patents. The Queen patents, for example, dominated that type of approach. So we wanted to use some new technology, and we used a method called framework grafting, which I talked a little bit about yesterday, where instead of taking the CDRs and putting it on a single human IgG backbone, we took a whole library of human frameworks, mixed them in with the CDRs, selected from thousands of variants, and came up with various hits that had the properties that we like, that worked very well in terms of being expressed and also bioactivity. And we screened those again, and you saw this data yesterday. So we came up with, through this framework grafting protocol, and using that novel technology, we came up with about 33 hits that were at least greater than 70% of the original activity. 12 of these hits were equal to or better than the wild type activity. The frameworks are all fully human. There were no substitutions in those. So they're going to be well behaved in terms of immunogenicity. And we converted this mouse molecule essentially into a human molecule by using this process. There were no back mutations, I said, that needed to restore binding activity. And you can see from some of the top hits, we actually improved affinity through the process. And this is something that's contralogical with what normally happens during humanization, where you typically lose about 10-fold affinity. And then you have to do substitution, either directed evolution or random mutagenesis to try to restore activity, and that's where you pick up some of these changes that can lead to immunogenicity or other problems with the immunoglobulin backbone. So this is our strategy. We want to show you just really the whole overall strategy so you can see what went on. So the human anti-CD22 mouse antibody LL2, which had affinity about 20 nanomolar, through affinity maturation, we came up with a clone that was improved about 20-fold, 1 nanomolar. We went through our humanization and characterization and selection process, and we came up with VM101, which had about a 200 picomolar activity. So overall, from the starting antibody, about an 80-fold improvement in affinity. And this matters in this case. This matters in terms of dose level and dose frequency in the human body during treatment and clinical trials. So here's a very important kind of assessment. When we look at EMAB, which is really the kind of starting antibody versus VM101, we did confocal microscopy using directly labeled immunofluorescent forms of either antibody. And then we could look at the relative mean fluorescence, and that's shown up there in the top left. And we got about basically a 10 to 20-fold improvement in surface binding to the cell through this process. So it was not just affinity selection. We actually went through a selection process where we looked at this binding to surface and then subsequent internalization. And the bottom panel shows you what the internalization looks like. So you can actually take a labeled form of the antibody, bind it to cells at 4 degrees, and then raise the temperature to 37 degrees and look at the rate of internalization. And after 30 minutes, you can see the difference between EMAB surface binding and internalization relative to VM101 surface binding and internalization. It was very rapidly internalized. And when we look carefully, I'll just move the thing around, you can see the actual localization. It was in a vesicle, the Golgi apparatus and endosome pathway that was just proximal to the nucleus. And this is where we came up really with the concept that we should be looking at something that's released and then goes into the nucleus and binds DNA. And that would be a mechanism that's separate from what Genentech was developing as a microtubule inhibitor, which frequently become resistant or get exploited by different pathways, and use a molecule that didn't have those properties, such that if there was relapse or resistance to the Genentech molecule, we would have a population that we can go into. So again, using the strategy about what technology is out there, we can try to steer towards something that we think is actually much differentiated than the molecules in the clinic. And this is part of this whole bio-better approach to protein and drug engineering. We tested actually the antibody just to see if it was working the same way that EMAB and LL22 did. And in fact, upon surface binding, one of our lead compounds did induce tyrosine autophosphorylation. It's not clear if that was actually really very much needed, but we went ahead and looked anyway. So we think we restored or recouped some of the initial biological activity that was associated with the starting antibody. So here's a little bit of the analysis, just at the end, our kind of a comparison of LL2, EMAB, the humanized variant of LL2, and then the different VM series that were, again, variants of mouse LL2 that have been humanized. And by and large, you could see that there was a correlation between increased affinity and surface binding and internalization, but it wasn't always the case. There are some examples of one, for example, VM18, which had a relatively low affinity, but it had very high surface binding and internalization. So even though we thought high affinity was leading to higher increased surface binding internalization, that wasn't always the case. So doing these type of functional assays were very important. And VM43, which actually became VM101, or our lead candidate molecule, consistently expressed at a higher level, lacked oxidizable residues, had a very good internalization rate and surface binding, and it was well behaved during manufacturing. So this is our candidate. We're going forward with that. We make that decision and we go forward. But we did have a couple of backups just in case something happened and there was some problem with it. The interesting thing is when you really look in alignments of the sequence of the proteins between EMAB and VM43, they're very highly related, but they're different enough that they represent a new composition matter in IP. And this was actually born out of our initial frame and operate analysis, and eventually we validated that by patenting and having those patents accepted. So let's go a little bit more into detail in terms of the actual construction of the ADC component. Again, I told you we had a concept where we wanted a typical type of payload. We wanted the newest generation of potent payloads that are out there, the best linker chemistry that we could use to get stability, and we're going to merge those all together and test them both in vitro and also in vivo, which unfortunately I left off on that slide. So the payloads we used were actually a family called pyrrolobenzodiazepines, or PBDs. These are molecules that can actually cross-link DNA together at very site-specific matter within the genome. And the interesting thing about these is, well, first of all, I'll mention these really came from the natural products derived from Streptomyces species. They were actually dimerized together so you can cross-link DNA in a specific way. What was interesting about that, they didn't cause any torsional distortion of the DNA molecule, so it was not repaired using typical synthetic mechanisms. The synthetic PBD dimers cross-linked and had very select activity in tumor cells versus normal cells. A lot of tumor cells lack mismatch repair enzymes that could take this out and repair it. So there was actually a specificity to get better activity in tumor cells than that. They're highly potent. They were more than 10,000 times more potent than the maytansinoids and the auristatins E's, for example, kind of a second generation form of payloads used by Seattle Genetics and things. And these were microtubulin inhibitors. There didn't appear to be any cross resistance with some of the standard chemotherapies that were being used in this field. And the other thing is that a naked version of this molecule, unconjugated, had already been in clinical development and it's shown to be more or less safety and efficacy in ovarian cancer patients. And in terms of that, with the FDA, there was a whole drug master file associated with that, so we didn't have to go with something that's novel, and that would really decrease our safety and toxicology studies that were part of the packet necessary to file an IND. So all of these things looked really good. So we created those. First we made the antibody and purified it. This is one particular run where antibody looks pretty good as a single peak. There was a little bit of degradation and aggregation. This turns out to be one particular run of that. But upon conjugation and cleaning that up using size exclusion chromatography, you can see you get a very single peak in species with no aggregation. So this had good manufacturing not only on the antibody side, the biologic side, but also on the chemistry side. So we knew early on that we were going to be able to develop a process that was scalable enough to make material in the clinical setting and also manufacture this as a product for market. So all of these things happened very early on before, again, spending a lot of time and money. When you looked at VM101 ADCs compared to EMAB in vitro, it had about 100-fold more activity in cell killing. And it killed a variety of lymphoma cell lines, some which are actually known to be resistant to rituximab. So we were very interesting to see those type of results, and some of those results panned out to in vivo studies I'll show you again. So all this looked very good. We liked this. These were early generation kind of linker drug combinations. There was going to be a lot of iterations on exactly how you do that, where we're going to click it into a certain place for the antibody molecule, et cetera. Those are all be determined, but right now, up to these studies, everything looked very good, and our earlier concept about engineering the drug to have improved potency in vitro binding actually panned out when we used the drug conjugate technology in vitro too. So all of that was working, and we were on par. So in terms of preclinical development, well, one of the first things we did was look at this in an animal xenograft model. And we used a particular model Dowdy experiment for a couple of reasons. First of all, had a moderately high level of CD22 on its surface. And in addition, this particular model in a xenograft study, it's been very hard to get complete really cell killing by a number of molecules that have been tested in this. We used a contract manufacturing group, contract research organization, the Piedmont group, part of Charles River. They're very reputable in understanding which were the best molecules. So we talked to them and said, okay, we have a potential antibody conjugate for lymphomas. We want you to basically provide us the information about the best model. We designed an experiment around that. We shipped them the drug, and they ran the model, and we got the results and the report associated with that. So how this model is run, we use again, the Dowdy-Burkitt, human Burkitt lymphoma cell line, a non-Hodgkin lymphoma cell line, and we put them in and xenografted them, let the tumors establish to a certain size, and then we dose once a week, every 3 weeks, with a number of compounds. We use the VM101 naked antibody just formulated in PBS. We use the VM1 ADC in various concentrations formulated PBS. And then we use rituximab. This is a marketed drug. We went to the pharmacy, bought some of it, and used it right in this model too, because this was really the competitor control arm of the study. So we really wanted to see how we compared to this in the clinic. And of course, we ran vehicle in the studies. And here's just a chart showing you some of the responses. I won't go on to this, but one thing we just saw that just stood out, at several different concentrations, we got complete response. We actually got not just stopping tumor growth, we got complete regression in the growth in 10 out of 10 of the animals studied. So it's very highly statistically significant. Rituximab worked very well, as you can imagine. But even in that arm, at 20 milligrams per kilogram, a very high dose, we were dosing from about 2 milligrams per kilogram down to 0.1 milligram per kilogram. So there's like at least a 2-log superiority in terms of the actual amount of material we needed to get complete killing. And again, we saw this. And we've repeatedly seen this. We actually have done this now in single doses, and we've just seen phenomenal results. This is actually what the xenograft model looks like, and you can graphically see the difference here in the top. This is just the growth curve measuring tumor volume over time. We carried this out to 60 days to see how durable this is, and then we compared that with the Kaplan-Meier chart, which really shows more or less the decay of the animals or death of the animals over time. And you may not be able to see it here, but at the 0.5 and 2.5 milligram per kilogram, these animals basically essentially had complete regression. There were no tumors in 10 out of the 10 animals. And this is the only thing we found in this study. The 0.1 milligram per kilogram, all the animals were still alive at the end of the study, but you could start to see a couple of the animals that the tumors were starting to get. So we were at a range where we start to see that we've reached kind of a minimal effective dose and also a maximum tolerated dose. In all of these animals, you could see that there was really no change in body weight. This is well tolerated. There were no toxicities associated with that. So this initial animal xenograft model said, okay, this looks very good. We now have a potential drug for development. And the rituximab arm, again, very potent, but you could see here one of the animals did die over time. And after a period of time, we expected some of those to be resistant.
How big were the tumors?
The tumors are about 150 millimeter cubed upon initiation, which is around what you want to be at, somewhere between 100 and 200 millimeter. Okay. So certainly palpable, and they basically went away. So just to summarize, we did a pilot test of the effects of V101 xenograft model. It wasn't even fully optimized as a construct yet. And we talked to the people at Charles River and said, okay, how does that look? And they said they have not really seen anything like that in these models for all the compounds we've looked at, small molecules and large molecules in the clinic. And they were very excited to understand more about that. The lead clone and the backup clone, they were assessed for manufacturability. They all looked good. We were very comfortable. We had clinical leads. I didn't show you the data, but we did some initial PK analysis, and it looked very good. It's what you expect to see for a human IgG1 in a rodent species. The chemistry and the production now is underway. The cell line developments are the next step. I'll talk about that. But we've recently found also some literature suggesting CD22 is expressed in certain lung cancers. And there may be uses between this that go outside just non-Hodgkin lymphoma, and we're starting to pursue that obviously. But as you'll get to in a minute, we got to a particular point where we had used our monies and we had to make some strategic decisions about how we're going to go forward with this program. We didn't own the conjugation chemistry. That was proprietary to another group. Even though we could utilize it to do these type of studies, we needed to make some type of partnership or deal. The next steps are going to be a lot of money. How we're going to finance that and take it forward, they're very important. So there were some decisions to be made. So let me just shift gears for a little bit and just show you some insights on what it takes to actually develop this. I know this might be a little small to see. You'll be able to look at the packets, but there's several points that you go through with milestones. And in the first part in green, you're really talking about making the antibody and engineering that. I'm sorry, I'm making David Eisenberg walk to the front here. That's, I'm grieving about that. I apologize for that. Okay, but if you look roughly what it takes to make an antibody and engineer it, and the time it takes, it takes you really better part of a year and maybe up to $500,000, $500,000 to do that. We did this a little bit cheaper, and that's all the engineering. That's outsourcing the work. That's doing the PK and manufacturing it. It can cost more. You may get it less, but to get to a human clinical candidate, that's about how much it costs, okay? And then the next steps, and there's a lot of value associated with that. I'll show you in a minute. When you go ahead and get it to that particular point, you've massively improved the value of that program. So I look at it, that's a minor expense to get to something that's a potential drug candidate and ready for clinical development, or at least for manufacturing. And then you have the next steps, which are the manufacturing steps and the IND enabling toxicology. These takes a lot of money. This is probably going to cost you about $5 million to get through that particular point, plus the safety, et cetera. And then there's the downstream things you need to do, writing the INDs, getting everything put over. So overall, getting probably through phase 1 to where point efficacy in the clinic, it probably takes you a little bit about between $8 and $9 million, so that's a lot of money, okay? Now we'll talk in a second about where the value inflection points are, and certainly there's a huge value inflection point in just getting to this type of preclinical efficacy, okay? Most people realize that antibodies are generally going to be safe and well tolerated in phase 1 clinical development. So you go from a point where you've massively raised inflection of the value, you've not spent so much money, okay? And then you go and you spend a lot of money and you haven't really changed the value of the program because you don't know if there's proof of concept in the clinic yet. So either stop there and partner or license, or you get the money to finance it and you take it all the way through proof of concept. And then there's another huge inflection value point along with that. So let's talk about manufacturing just a second, and I'm not going to go into this in much detail. It's a very difficult and expensive process. This is not just throwing a vector into 293 cells, making some medium protein A chromatography you can do in the laboratory. This is a process of actually generating a cell line using vectors that are going to give you very high level of expression, and generating a cell bank that's uniform with respect to integration of that vector and stable, free of viruses and other pathogens that might be present there. And you've produced a master bank that's possible to then take vials, manufacture this drug by expanding them over the lifetime of the cells of this. So that's a very complicated process just for the cell line development, et cetera. And then there's the process development, taking the media from that and coming up with a very streamlined process for manufacturing. You usually like to be that 3 steps to where you get your final material. And that has to all be developed and be done in a way such that every time you take a vial out and go through this process, you're going to get something that looks the same every time, and you can reproduce that, and you can demonstrate to the FDA that you've done it that way. And then there's the actual fermentation, the bioreactor, the generating the material to make clinical material. And we're talking about kilograms of material, not just a few milligrams or something like that. You need to make a large supply. You have to test the stability and show over time that it's stable and it's going to look the same time. So this is a very rigorous process. And doing this in the most streamlined, quickest way takes about 12 months. So if it's taken you a year to make the antibody, 12 months to do that, it's about, you're about 24 months from the time the light bulb goes off with the drug concept to getting ready to enter the clinic. And then you have to take that material, of course, and do safety studies. You should also know that this is mostly done at contract manufacturing unless you're a very large pharma and you have this internal thing. This is not something that you want to start, and as you're growing a company, for example, I'm going to set up a manufacturing process in my laboratory and go and do that. It's just the wrong thing. It's a margin business. You don't want to do it. There's professionals out there. You work with them as a contract manufacturing organization. And there's great people out there, Boehringer Ingelheim, Lonza, for example. Those are probably the 2 premier CMOs. And when you use them, they'll pretty much guarantee you're going to get in the range of about 5 grams per liter. And that's an incredibly high amount of protein per liter if you really think about it, 5 grams per liter, where almost a single product in the media is a recombinant monoclonal antibody. If you look at what this takes also, I'm not going to go into timelines and Gantt charts, but if you look at this process, this is a little segment of a timeline that shows going from starting the cell line development to filing an IND for 12 months. And there's about 1,500 lines of high-level rolled-up processes. They're very tightly coupled, and the critical paths are very difficult. And I don't want to go in to show you that, but just to say, again, this is something that you want professionals to weigh in and do for the people that are experienced. You want to have people that are managed the whole CMC process, which is chemistry, manufacturing, and control, and help you shepherd through that and all the auditing that needs to go on. And this is for the front piece of the manufacturing. You get to the point where you actually do the IND enabling studies, which are the safety and tox studies, which usually you'll do a safety and tox study of about 30 days because this is about how one dose is going to last and things like that. And you have to do them as single doses and, of course, repeat doses. Do these normally in non-human primates for antibodies because of the cross-reactivity and the pharmacology of antibodies. So these are very expensive studies and detailed studies that go on. Again, I'm not going to go into the timelines about that, but I want to expose that to you again, that this is a process that as you go from I've selected a clinical candidate to filing IND, that's very complicated. You need experienced people that understand the timing and how to do this as quickly as possible and try to make as few errors to not only get it done to the safety studies, but have it be acceptable by the FDA as a new investigative drug for clinical testing, IND. So going back to, again, the dilemma we had, going back to the same timeline, we knew that we basically either go out and raise a lot of money to take this all the way to proof of concept. We're going to have to license the technology. We're going to need more money for that. What can we do at this particular time? We realized there's a few places to exit. At preclinical stage, we could sell the asset or partner it. Those are possibilities too, because we have very good activity. We know we're going to be able to go through this whole process. We can exit it at the IND, or we can exit at the proof of concept in clinical studies. We felt there were two exit points, exit at preclinical or the clinical proof of concept, because at the exit IND, you haven't substantially increased the value of the program from where it is at the preclinical stages. You spent a lot of money, you made a cell line, you can manufacture, et cetera, but you haven't really substantially increased the value. So if you're going to go through the manufacturing processes, the steps in yellow, blue, and red, et cetera, you better be ready to take it all the way into the clinic, because there can be huge value inflections. And here's one of the reasons why, and this is actually a chart of some of the deals that have been made, usually over the last 10 years. And you can see things that have been partnered at various stages by small companies shown there in the very right-hand column. The target for those particular antibodies were shown. And then the eventual partner, which are all very large companies looking to satisfy their pipeline needs and gaps for new drugs that are coming out of. And you could see some of the upfront cash associated with those. There's some pretty big numbers. So looking at some of the preclinical stage studies, for example, the VO, basically antibody to RON that was taken by J&J Centocor at the preclinical stage, $15 million upfront payment for that, okay? Lifetime with milestones worth up to $555 million should it go into actually the market. And there were net royalties on the sales, which are not shown here. So these were very valuable assets at the preclinical stage, okay? If you take them on forward, you can see some of these deals, for example, at proof of concept, the Micromet Epicam deal with Serono, upfront payments almost to $150 million, and then, of course, downstream royalties and milestones. Some of those are not shown and not publicly available. So we really, just going back to this, we looked at this and said, okay, let's do a deal at preclinical stage. Let's get some upfront cash. Let's go ahead and get some milestones and royalties in that, and then some basically royalties in the future going on to fund that. We're going to take these funds and put them into other programs and use this as a sustainable model to build more molecules and put them forward like that. And eventually we're going to accumulate the funding to be able to take something all the way through phase 2 and build value. So this is actually ongoing, this sustainable model where you make a single investment of about $500,000, you try to get some upfront payments here, and then you put those back into development of your other molecules. So we were developing a pipeline that over time we're going to get to the point where we'll build higher value, leverage that, build the organization, and go further ahead. And that's what we did. We formed a partnership. This was announced last June, where we actually did a deal with a company, ADC Therapeutics. They have the technology available, the PBDs, or the pyrrolobenzodiazepines, very potent linkers. Can't tell you too much about the deal here, but it did involve basically taking over the asset by ADC Therapeutics as a joint partnership. So we still own part of the molecule, but we got some upfront cash. We had downstream royalties and milestones and royalties due, but we still owned a lot of the molecule, okay? And if we wanted to own more of it and share up to 50-50 of it, we could put some cash in to help development. So this is really a way of taking this forward, solving our problem on how we're going to get access to the technology, because they have the technology, the linker and the conjugate. We have the antibodies, so we merged in that type of partnership going forward. And this molecule is actually going on into development as we speak right now, going forward as part of a portfolio of ADCs, very potent, next generation, best in class linker conjugates, therapeutic antibody drugs. And that shows you really, in essence, if you look at it, what the strategy was on how to get there in the time. And I think that incorporates a lot of what we talked about in terms of use of the technology, unmet need in the clinic, how you actually go through the process of making the antibodies, testing them, moving them on through the process of preclinical development, IND enabling things, manufacturing, all these things, wrapped up into the strategy of how you do this. And in a world where the macroeconomics for small startup companies was very poor, and we didn't want to leverage too much of the company outside by taking in venture capital. The last thing I just really wanted to touch on here is intellectual property. How does this strategy fit into all of that? We didn't really talk about that too much, but all along this whole process, you're thinking about generating IP. And here are some of the main areas that are inserted across this timeline on when you want to start to develop your IP. Up front, if you have a novel target, you'd like to say, I want to get sequence composition on the target and generic claims to antibodies. This is very tough to do nowadays, but it used to be, I cloned a gene product, I'm going to say, I automatically claim antibodies to it, even before you've actually made the antibodies per se. This was a standard approach to try to get generic antibody claims, and then you could later on actually file patents downstream on specific antibodies to that. There was a freedom to operate assessment done right up front. Okay, can we work in this area? Don't go to laboratory, don't spend any money, work with general counsel, IP counsel, et cetera, and determine, is this a way you can actually go forward? And if you do, let's go ahead and do that, but let's have a very clear strategy on how we're going to navigate through that. Then you can actually make antibody and say, these antibodies, and a list of them, I'm showing CDR sequences basically, and claiming those and bind to the target, showing the biological activity associated with that. That leads to a set of claims around the method of use of these antibodies. I'm going to use these specific antibodies to treat these diseases, and you can start to broadly say, now I'm starting to generate a position where novel antibody drug conjugates to treat these diseases and start to basically build a more or less a defensive area around the place you want to operate in. And then eventually you want to file the sequence on the actual clinical composition. And you notice that comes latest, okay? You might say, well, why wouldn't you do that immediately upon getting the antibody? We have to think about patent life. And what you really need is composition of matter on your drug, the actual sequence of that. And if you file that too early, you're burning patent life. If you think about some of these drugs, these may sell $1 billion a year. 2 years of extended patent life, that's a lot of money. You have to think about when you're going to file that sequence. So using the technologies I talked about yesterday, you can get up to 10 to the 9th different variants, et cetera, like that. It's highly unlikely, almost impossible, that someone is going to come up with the same antibody sequence you have and be able to claim that, okay, before you do that. So you'd like to hold off until the last minute possible before you file that patent. In large pharma companies, this is done right at the point you file an IND, because this is actually when you're transferring the material out to the company, to other places where someone could actually take that and be able to sequence it and file patents on it. In smaller companies, you do that right about the time you transfer the sequence to a contract manufacturing group, or you do a partnership, et cetera. So you do it much earlier, but you want to do it as late as possible. And of course, I should mention that, you know, when you're working with a company or you're working with a university, you're working with people that are developing an IP portfolio, and it's usually the company or the university that actually owns the IP, so they're managing it in a way that's very important to defending the area and making sure that people want to come in and license, they have all the proper patents together to increase the value. So I'm looking at the clock and the time. I think I'm going to wind down here and just tell you a few takeaway points here. I'm not going to read over this particular, but over the last two lectures, we've talked about a few different things. We've talked about the area of therapeutic monoclonal antibodies. This is a very important area, fastest growing source of new medicines, and some of them are very lucrative blockbuster products. In fact, the largest selling medicine of all drugs, whether it's a statin or something for depression, et cetera, blood pressure, is actually a biologic. It's a Humira for the treatment of various autoimmune diseases. There's a diverse number of targets, but that target list is expanding as we're getting into greater novelty. Remember that of the 3,000 secreted proteins out there, we really only prosecuted about 100 of them in terms of drugs. So there's a huge number of things that need to be done to validate some of these others so we can go after novelty for treatment of all types of diseases. Some of these classes are going to be easy to get antibodies to. Some of them are relatively intractable. So there's a continual development of new technology to try to get antibodies to some of these intractable things like GPCRs or ion channels, which are very druggable from the small molecule side, but have not yet been very druggable but from the antibody side. There's multiple types of antibodies. They come in all shapes and flavors, large, full-length, bivalent human IgGs. There's small fragments. There's small fragments stuck together, et cetera. There's antibodies from different species, camels, sharks, et cetera. They're all being used in clinical development right now to expand the type of modalities that use them, give freedom to operate for new medicines, et cetera. The pharmacology, pharmacodynamics are very important. And you can control those in a variety of ways, some through the shape of the modality, some through by optimizing the variable domain and its interaction with the antigen. And there are these other properties that, you know, maybe don't seem so science-based, but extremely important: the manufacturability, the IP, you know, the integration of all our target biology to come up with a new drug concept, and your strategy on how you're going to take these all together and navigate through the clinic. So with that in mind, I'm going to end the lecture here. I just want to make some acknowledgments. These have been mentioned before, of course, but thank you very much for the Department of Pharmacology for inviting me to come lecture to you guys. We've been working with UCLA, the CTSI, and also within UC Braid, the CAI, or the Center for Advanced Innovations. This is part of the NCAI, which involves the integration of UC Braid, Harvard, and the Ohio clinics together as a group where we're really working together to expand basically the integration of kind of the development of internal assets within the university for commercial products. And there's actually a whole sector of skills development from which part of this class is being added on to, where we're actually teaching various things, whether it goes from how to make drugs, how you look at IP, et cetera, how do you teach this and go forward. And then, of course, there's the National Heart, Lung, and Blood Institute, which all this work is being funded through. I want to just mention also there's an upcoming webinar that to continue this series. If there's interest, you can go to our website here, and these are all being scheduled. These are being captured, and people are able to get the actual PDF files of the presentations, but also hear the talks as well. So thank you very much again, and I'm here to take questions, both from the audience here and for those that are online now.